Welcome back to another explainer. Let's just dive right in, because every single living thing on this planet faces one massive, undeniable biological challenge: species survival. I mean, how do we keep going? How does life actually persist generation after generation in a world that is constantly changing around us? Well, today, we're unpacking the mechanics behind nature's ultimate survival strategy. It really begs the ultimate question, doesn't it? How do organisms cheat death to maintain their species? Because, let's face it, individually, every single living thing eventually reaches the end of its lifespan. The evolutionary solution, the ultimate workaround to this seemingly unavoidable problem, is the biological imperative of reproduction. So, reproduction. By definition, it's the production of young ones like the parents. It is absolutely crucial because it leads to the continuation of a species, and really it maintains the overall continuity of life on Earth. You could think of it as nature's prime directive. But what's incredibly fascinating is that this baseline survival instinct is achieved in wildly different ways depending on the organism we're looking at.
Which brings us to Section 1, the Great Divide. Choosing a path. All these particular methods of reproduction generally fall into two main categories.
We generally see two distinct pathways here. On one side, you have sexual reproduction, which involves the fusion of compatible gametes or sex cells. But on the flip side, we have asexual reproduction, and this completely skips that fusion process. There is literally no partner required. And for today's explainer, we're going to focus entirely on this highly efficient, incredibly self-reliant, asexual route.
Moving right along into Section 2, enter the clones, nature's perfect copies. Let's look at the specific results of taking this asexual route.
Asexual reproduction produces genetically identical progeny from a single organism. Think about that for a second. A single parent, just one, is all it takes. That one organism passes down its exact genes to the next generation. There's no mixing of traits, no matching up of different genetic codes. It is just pure, unadulterated duplication coming straight from a single source. And because of this exact duplication, the resulting individuals are called clones. They are both morphologically and genetically identical, meaning they don't just look exactly the same on the outside, which is the morphological part, but they inherit and share the exact same biological code on the inside. It's a true biological copy-paste. Here in Section 3, we're opening up the Asexual Toolkit, Methods of Cloning. Organisms choose to reproduce asexually in a few different ways, utilizing specialized tactics to ensure they don't just survive, but actively multiply.
Nature relies on a sort of cheat sheet with three primary survival tactics here. First up is fragmentation, where pieces of a multicellular organism grow into completely new individuals, like we see in the algae Spirogyra. Then there's budding, which relies on outgrowths that separate to develop, and this is incredibly common in yeast. And finally, spore formation. This utilizes flagellated, motile zoospores capable of independent growth, which happens to be the go-to method for Clematomonas. Let's break a couple of these down further to really understand the mechanics.
Let's zoom right in on fragmentation. A multicellular organism breaks into fragments. Now, you might think breaking apart sounds like a tragic end, right? But actually, it's where the survival magic happens. These fragments regrow into entirely new individuals. For an organism like Spirogyra, breaking apart is literally a new beginning. It's an automatic, built-in survival tactic triggered by environmental forces.
Now moving on to budding. This one is all about thriving in favorable conditions. It's the most common method in unicellular organisms like Protoceathon and yeast. In fact, when the environment is warm and food is plentiful, yeast can double its entire population in just a couple of hours. The organism rapidly produces these outgrowths, or buds. Once these buds mature, they just pop off, separating from the parent to become entirely independent, fully functioning individuals.
Okay, Section 4. Microscopic Survival Strategies, the Expansion Pack. Because beyond fragmentation, budding, and spores, there's a whole other tier of specialized methods organisms use to clone themselves.
The sheer diversity of these ancient tactics is honestly staggering. We see binary fission in organisms like chlorella, diatoms, and chlamydomonas, where they're literally splitting themselves completely in half to multiply. We see conidia formation acting as a highly effective method for penicillium. And yes, that is the exact same fungus that gives us penicillin. And then there's gyma formation, which is utilized by the liverwort Marchantia. These are highly adapted tools, ensuring life finds a way to persist without ever needing a partner.
To wrap up this explainer, I want to leave you with a slightly provocative question. If a clone inherits the exact genes of its parent, is it truly a new individual, or is it just an extension of the original life? It's a fascinating philosophical puzzle wrapped right inside a biological reality. Think about that the next time you see a patch of algae or use yeast to bake some bread. Thanks for joining me for this explainer. Keep asking great questions, and I'll catch you next time.